Hello everyone, welcome to my channel, Chorea D Antigen. So we're going to talk about monohybrid inheritance. This is our second video about genetics, monohybrid inheritance. So monohybrid inheritance is a one-trait inheritance, and Mendel performed crossbreeding experiments to discover this, and he used true breeding homozygous plants and chose varieties that differed only in one trait, that is monohybrid cross. So he performed reciprocal crosses, and that is if he used the parental generations. For example, if he crossed tall plants with short plants, and he got a certain generation, maybe female tall plants and male short plants, after that crossing, he got the results. He discovered that they're the same results he gets after crossing male now tall plants and female short plants. So he discovered that things like height of the plant or the organism is not sex linked. So he performed reciprocal crosses, and then he formulated the law of segregation, which we will look at in further detail as the video later goes on. So we're going to look at this example of the monohybrid crosses. So we're seeing the parent generation, yes, which shows that they are the tall plants right there being crossed with the short plants, and there is formation of the gametes right there. And then using the Punnett square, we're able to see that the offsprings are 3 in a ratio of 3:1, where most of them, like 3/4 of them, are tall, and there is a 25% chance of having a short plant. But then, as you look at that thing in detail, we'll have dominant. We have 25% chance of having one homozygous dominant tall plant, and 25% chance of having homozygous short plant, and the rest are just intermediates with their heterozygous tall plants. So we have the law of segregation, which I told you about earlier. So we're going to look at this law of segregation. So in the law of segregation, we see that each individual has a pair of factors for each trait. Now these factors are called alleles. Now the factors, or another one would say alleles, segregate or separate during gamete formation. So we're seeing the second aspect, saying that these factors separate during gamete formation, and each gamete contains only one factor from each pair. Only one factor from each pair. So each gamete will contain only one factor from each pair, like we saw right here. If you look at this, yes, there's supposed to be 2 capital T's, and now you can see that there's only one factor goes into the gamete. So we're seeing only one small t in the circle. That is the representation of the gamete, only one factor. And then the fourth thing is that fertilization gives the offspring 2 factors for each trait. So the offspring, if you see the offsprings right here in the Punnett square, we see the offspring having one factor from each side, because this capital T was homozygous dominant for the tall trait, the trait, and then we're seeing there are some offsprings here having a capital T and a small a lowercase t. So that one shows that this offspring obtained one factor from the mother, and maybe from one gamete and another from another gamete. Now in modern genetics, we have a way we look at this monohybrid inheritance. So we see that each trait in a pea plant is controlled by 2 alleles. These alleles, for those who don't know, are the alternate forms of a gene. Now the dominant allele, which is the capital letter we looked at earlier, masks the expression of the recessive allele, which is the lowercase. Alleles occur on homologous pairs of the chromosome at the particular gene locus. So homologous pairs, these homologous pairs of chromosomes would be two chromosomes that are paired together, but these chromosomes may have the same shape, the same size, or the same structure, and then they have the same genes at particular positions or locus on them. So these genes can have two different forms, alternate forms. Can have one form and then another form. So those are the alleles on the genes. But the homologous pairs have the same. So these homologous pairs of chromosomes, one chromosome maybe on the right can have a dominant allele, and then the other can also have the dominant allele. So it can have capital T in the same position, yes, or we can have a capital T, small t on the other chromosome. But these chromosomes are homologous. So the homozygous, this term, we're going to look at it deeper in genetics, but we need to know that this term means identical alleles. If we say homozygous, the alleles are homozygous, meaning they're identical. Then if you say the genotype is heterozygous, meaning that these different alleles, these alleles are different. If you say that the genotype is heterozygous, so if the genotype to be homozygous, then the alleles have to be identical. For example, a lowercase t. Then for heterozygous, it should be a uppercase T or capital T and the lowercase t. So that genotype would be heterozygous. Now for homologous chromosomes, this is what I was describing earlier. Yes, we're seeing that showing us a diagram of homologous chromosomes showing that they have various alleles at specific loci. So various, we can see the capital G and the small g, the side, same positions, they're similar structure and similar genes. Yes, similar genes. The gene maybe G can be for let's say height, then the other small g for still height but small, the shorter version. Then we can have them having the same, this called sister chromatids, are having the same alleles on the loci. We can have a capital G right there and a capital G right there. So genotype versus phenotype. A genotype refers to the alleles of an individual for a specific trait. So refers to two alleles of an individual for a specific trait. That is a genotype. Now if identical, these alleles, the genotype is homozygous. If different, the genotype is heterozygous. Now for the phenotype, is that this refers to the physical appearance of the individual. This one is also called the outward expression of the genes, outward expression of the genotype. For example, if we have capital T, the homozygous tall plant, homozygous tall genotype as homozygous tall, we have the outward expression is a tall plant. You get what I'm saying? So that's the phenotype, what we can see with our own eyes. Then for a Punnett square, in a Punnett square, which I talked about earlier, the other box. So it's a table listing all possible genotypes resulting from a cross. All possible male genotypes are lined up on one side, and then all possible egg genotypes are lined up on the other sides. And every possible zygote genotype is placed within the squares. This is what I'm describing. So if you have a look at it, you'll see we're using an example of the earlobe inheritance patterns. So we have members of the population having ears that are attached to the skin there. They're like this. You know what I'm saying? Very attached. And we have those that are loosely. They're loose. They're not attached to the other side of the skin. You get what I'm saying? So we have those ones. So we have that example right there. So the Punnett square is that table that you see right there. So we have, are you seeing the eggs right there? Then the sperms this side. Then the possible offsprings are right there in the middle. You see in the capital E, capital E. We are seeing those, and then the small e. So those are possible. Yes, phenotypes, and the key is right there. Yes, if you want to dive deeper into it, that is video magic. You can just pause and do what you want to do. So we're going to look at the test cross. In a monohybrid test cross is used to determine if an individual with the dominant genotype is homozygous dominant or heterozygous for a particular trait. So here we're trying to figure out whether the individual that we are seeing appearing tall, I'm using the example of the height, appearing to be a tall plant, is homozygous dominant, that is having both alleles for having two dominant alleles for the height, or being heterozygous for that particular trait, for example height, having the one allele for one dominant allele for height and one recessive allele for height combined together. Though this individual might appear tall, but it's possible for having a tall person by having a recessive allele on the chromosome. So we have the monohybrid test cross. Individuals with recessive phenotype always have heterozygous recessive genotype. So that is key. For a person to be recessive, having a recessive phenotype, for a plant to be short, completely short, that means, or significantly means, that they have homozygous recessive genotype. Then, however, for individuals with dominant phenotype have intermediate genotype, may be homozygous dominant or heterozygous. For a test cross, it can be done to determine whether the genotype of the individual is dominant, to determine the genotype of a dominant phenotype, whether homozygous or heterozygous. Now, for one trait test cross, where the unknown individual is heterozygous, I want to blindfold your mind. I want you to think like this individual. You don't know that this individual is heterozygous dominant, heterozygous for a particular trait. So you just cross the capital T, small t, with the lowercase t, and then you cross, you get that, figure out that the phenotypic ratio is 1:1, having some members tall and others short. So that one will show that the dominant maybe the dominant phenotype is heterozygous, has an heterozygous genotype. That will show that the dominant phenotype has an heterozygous genotype, because we are having half the offspring tall and the other half short, in which is a ratio of 1:1. So remember that is, if you want to test this, you can only do this if you cross that unknown dominant phenotype with a known homozygous recessive phenotype. You get what I'm saying? So that means you get that tall plant you want to figure out, and you cross it with a very short plant, and that is the only way you can figure this out. So if you get that 1:1 ratio, meaning that dominant individual, or that dominant phenotype, that tall plant, or that tall plant has a genotype which is heterozygous. Now, for one trait test cross, individuals, we're trying to check out whether the individual is homozygous. So if you cross that tall plant with a short plant, And then you end up having all the plants, all the offspring stored. So that will mean or imply that the individual, the tall individual, was homozygous. Homozygous. That is the only explanation that I can give. That's the only explanation I can give. The individual is homozygous, meaning is completely dominant, is homozygous dominant of that particular trait. Now we're going to look at monohybrid inheritance in humans. We have been looking at plants. Now we're going to dive deeper into some humans. Now we're going to begin with the genetics of tongue rolling. I think some of you know this, some of you don't know it, but some individuals of the human population are able to roll their tongues while others are unable to roll their tongues. I can give you an example. I'm a tongue roller, so let me show you. I think what I've just done with my tongue right there. It's video magic. I'm not repeating it. If you want to see it, you can pause and play back and see what I've just done there. So that is what we can see right there. What we can see right here. We can see that tongue, a rolled tongue, is a dominant. So the allele, or the allele for tongue rolling, is dominant over that one of the flat tongue for those individuals who are unable to roll their tongues. So meaning the other allele for a flat tongue is recessive, and as we can see right there, for every child there are possibilities. There are like 4 possibilities. So if you have an individual who has 2 alleles, the dominant alleles for rolling a tongue, that individual will be homozygous for tongue rolling. And if the individual has the recessive allele and the dominant allele all together, they will still roll their tongue. And still, that is it. And if that individual, you get an individual having flat tongue, the allele for flat tongue and the other allele is also for a flat tongue, so they will be homozygous for the inability to roll their tongue. So they will be non-rollers. So tongue rolling is inherited, and you can inherit it from your parents, maybe from your mother or your father. It depends on their genotype. So for this example, we're using capital R as the allele for a roller, and the lowercase r as the allele for a non-roller. And then when you punch it into the Punnett square right there, we can see that if you cross individuals who are heterozygous for tongue rolling, you end up with individuals who are tongue rollers in a ratio of 3 to 1, the one being taken by those who are non-rollers. We have the other 2, maybe the 50-50, half of the individuals being heterozygous, like their parents were, while the other half, the 25%, being dominant tongue rollers, while the others are non-rollers. Then we'll have monohybrid inheritance in humans again, but right now we're going to look at the Rhesus D Antigen. Now the Rhesus D, the Rhesus positive, you have had this. The doctors have told you immediately before the blood transfusion. Maybe they tell you, man, you're Rhesus positive, you're Rhesus negative. You've had this. It's all over the internet. So it's very good to know your position in this case. So in addition to the ABO blood system of antigens, most people have further antigen on the surface of their red blood cells, and this is called the antigen D. Yes, we have heard of antigen A, antigen B, antigen, no antigens. You get what I'm saying. All, but with the all antigens, we've had many individuals having both A and B antigens, those having none. And then there is also this one, the D antigen. So if it is present on the surface of red blood cells, you'll be called Rhesus positive. Then if it is absent on the surface of the red blood cells, you'll be called Rhesus negative. So it's represented by that capital R lowercase h plus, rhesus positive, then capital R lowercase h minus, that is rhesus negative. So those that are rhesus negatives don't possess this antigen, but these individuals react to the presence of the antigen D by forming anti-D antibodies. So the anti-D, this is a specific type of antibodies that are specifically produced by the body to fight against a specific antigen, the presence of an antigen D in the blood system. Now, for this continuation, we have that if a person is rhesus negative and gives blood to an individual that is given blood that contains rhesus positive antigen, blood that has blood cells that have the antigen D on their surfaces. So this individual respond by producing anti-D antibodies, and this leaves the person sensitized. So produce the antibodies, and these antibodies will fight against the what? The red blood, the antigen D. But if this individual specifically is given more red blood cells, more rhesus positive red blood cells, and during this blood transfusion they are given more of this individual might suffer from severe or fatal agglutination. Now in agglutination is that what happens in agglutination is that the red blood cells are caused to clump together. That all the red blood cells that have been blood transfused or have been put in this person that is negative will be forced to clump together, like you can see right there. You're seeing some red blood cells that are joining together, forming clumps or clusters. You get what I'm saying? So that is agglutination. So if this happens, if your blood agglutinates, you may end up with severe, you may even die, because just imagine if something blocks the water pipes. Something blocks the water pipes, or the water within the pipes freezes. Just imagine that if the water within the pipes freezes, meaning that there will be something that will be hindering the flow of water within the water pipes. So this is what happens in the red blood cells, in the blood vessels, that if these red blood cells agglutinate, they clump together. First of all, they're not going to carry out their function as red blood cells, and they're clamping together. They're going passing through the tiny capillaries of the body, so end up blocking somewhere, clotting. There'll be some kind of swelling. There'll be death. It happens in the brain. It's very, very, very deadly. You know what I'm saying? Very deadly. If these people can really die, it can cause death. It's a very bad thing. So you need to first know your status, your rhesus status. You need to know your rhesus status. So the presence of anti-D. So the antigen D, yes, the allele that causes an individual to have antigen D on the surface of red blood cells is a dominant allele, while that one that causes the individual known to have the antigen D on their red blood cells is a recessive allele. So we have those different crosses, as we can see right there. It's video magic. If you want to see it clearly, you want to review something, you can play back, pause, do whatever you want to do. So if you cross those individuals that are dominant right there with a recessive individual, will have individuals that will all be rhesus positive, like you can see right there, and then the other cross can be seen also. So I'm going to give you some examples of recessive monohybrid inheritance in humans. We have albinism, which is the inability of the body to make melanin. Yes, so if a body is unable to make melanin, this protein or something, the individual will turn out to be an albino. They will be very light-skinned without this melanin that makes me black. Yeah, shout out to all black people. They have a lot of melanin in their body. Yes, so we have cystic fibrosis is also disorder, genetic disorder of the mucus secreting glands. So these individuals will end up having thick mucus within their lungs, very thick mucus within their lungs. It's very deadly. Oh God, cystic fibrosis. So it's a chronic disease, but it's inherited. You inherit it either from your parents, grandparents, or whoever is in your lineage. We have phenylketonuria. So it's an inborn error of metabolism, also known as PKU. It's also a simple Mendelian recessive trait. So it's a recessive, it's not dominant. Yeah, you can, if you have the other dominant allele, then you will you won't be sick of this disease. But if you ever marry a person with the same allele, there's a chance that you may produce children that have this phenylketonuria. So it's also not a good thing to have, but for those individuals who have it, you're there, we're also there. Examples of dominant monohybrid inheritance in humans. So we have the Huntington's chorea. Huntington's chorea, it's a degeneration of the nervous system, which leads to premature death. So this is the dominant case that we're talking about. It's dominant. If you have it, you're gonna have it. If they have an allele having this, it's a dominant one. You're going to suffer from this. If you have it, you're going to suffer from it. But the good news is that you won't see its effects until you are in the mid-30s. I don't know whether it's good news or bad news, but it's determined by a dominant allele, and the allele is not expressed in phenotypically until you're about 38 years of age, when the sufferer probably have had a family or passed on the allele. So we're going to look at the genetics of Huntington's chorea. And so the H is representing the allele for Huntington's chorea, the presence of that allele. Then this lowercase h represents the allele for a normal condition. So there are 5 different combinations. We have the upper cases of H crossing each other. Yes, we have that one in the middle, then the second one, then the other one, then the other one. All of them right there, video magic. Then we have, so if you have crosses of homozygous dominant individuals with Huntington's chorea, the offsprings will all not survive. They will all have this trait, they will have this disease, genetic disorder. Then if you cross individuals, one individual is homozygous dominant and the other is heterozygous of the Huntington's chorea, the offspring, some of them will, still none of them will survive, you get what I'm saying, but one will be homozygous. But they won't survive, they will have the disease. Then if you cross those that are heterozygous, the heterozygous individuals, completely heterozygous, 75% will not survive, only the 25% will survive. And it's a chance that they'll survive. There's a possibility of not producing any survivor. Then we have Huntington's chorea. The genetics, more of it. We have, if you cross individuals that one is heterozygous of this condition, and the other is completely recessive of this trait, having 2 alleles, recessive alleles of this, so we'll have a 50% chance of survival, because some individuals will have it and others won't, but there's a 50%. But no one will know that they have this condition until they're in their mid-30s. That is the disadvantage of this disease. So it's very deadly disease, one of the most deadly among all that I could ever research about. This is the most deadly. So if you have it, pray to God. If ever discover that you have it in the mid-30s, I would advise you to pray to God, who is able to heal all diseases, known and unknown, to cure you from this disorder, because it will cause degeneration of the nervous system. And nervous system, we're talking about the central nervous system, talking about the neurons, talking about the brain right here. So just imagine if you're degenerating and it's all dying out slowly. You end up dying. A very painful day. Painful in terms of the nervous system. But thank you all for watching my videos. So you can subscribe for more if you want. If you don't want, I'm okay with it. You know what I'm saying? I'm okay with it. Yeah, I believe in the theory of those who want will subscribe, those who don't want won't subscribe. But just know that if you subscribe, you're doing me a favor. You're just helping me. The Ugandan right here. So you can comment, make your comments. If there are any mistakes that you've seen in the video, I'm all ears. You can just comment somewhere, write a comment and correction. Anything you want to add, put it in the comments. Any video you want me to make, maybe a video you want me to make about genetics of something, just put it in the comments. You get what I'm saying? If you just want to laugh, put emojis, put it in the comments. You get what I'm saying? So see you next time. Like and subscribe. See you in the next video or lecture 3. Bye bye.